Okay. So saya sambung So if you have any question, you can ask me. So saya tak faham lagi, sir. Kena ulang balik lah, sir. Can I proceed? Muhaimin?
Yes, sir. Can.
Can I proceed?
Yes.
Okay. So this situation, we can say that the parental phenotype combinations are more common than expected. Dia macam lebih cenderung lah nak ikut macam dia punya parental phenotype combination. Okay, remember this word, parental phenotype. Okay, so we can conclude this by looking at this data lah. Data ni lah yang menunjukkan bahawa okay, purple flower long pollen and red flower round pollen, okay, they show greater number compared to the expected. Yang kita expect 9. And then when we do the actual experiment, it gives the value of 15.6. This one is 1, the expected ratio. And then we get 4.5. Dia jadi makin tinggi lah. So dia menunjukkan lah bahawanya seolah-olahnya F2 ni dia macam intend to follow the parental phenotype combination. Okay, so what we learned from finding by Batterson and Punnett, okay, Batterson and Punnett discovered 2 traits that did not assort independently. Okay, dia nampak macam dia tak pergi secara rawak lah, benda ni macam ada setengahnya nak ikut-ikut, dia tak pergi secara rawak lah. Okay, they suggested that the transmission of the 2 traits from the parent was somehow coupled. Tak tahu, dia macam masa tu dia tak faham lagi lah. Dia kata macam somehow coupled, entah macam mana pergabungan tu berlaku kan. Okay, so the 2 traits are not easily assorted in an independent manner. Okay, however, they did not realize that the coupling is due to the linkage of the 2 genes on the same chromosome. Masa ni Batterson dengan Punnett ni dia tak dapatlah jawapan lagi masa tu lah. Dia tak dapat jawapan lagi. Tapi dia dapatlah something yang something that unusual from what they have get before. They find something that precious to be discovered more by other researcher. Okay. Okay, so today we learn about crossing over and recombinant. In diploid eukaryotic species, linkage can be altered during meiosis as a result of crossing over. Okay, crossing over occur during prophase 1 of meiosis. Okay, you have learned in the previous lecture. Replicated sister chromatic homologs associate as bivalent tetrad. Okay, cerita apa pula ni? Tiba-tiba bivalent pula ni. Okay, non-sister chromatic. Non-sister chromatic, still remember yang saya cerita tadi? Non-sister chromatic. Okay, non-sister chromatic of homolog chromosome exchange DNA segment. Okay, this one is from my mother, this one is from my father. So something happened between these 2 homolog chromosome. Okay, so we can see here there are certain protein developed. The name of the protein is synaptonymer complex. Start to form and synapsis begin. Synapsis tu bila dia jadi duduk dekat-dekat lah, dia tertarik. Okay, and then this form bila dia dah tertarik, we call as this position as bivalent. Okay, dia duduk setara keadaan dia. Okay, jadi kita tengok video. And then crossing over occur, berlalulah exchange tadi between non-sister chromatic. Okay, and then the point where the exchange happen, point ni kita panggil sebagai chiasma. Okay, Many points because kias mata. Okay. So this is an animation. Saya tunjuk dulu satu animasi ni. Okay, we have sorry. We have animation of crossing over. This is histochromatic. Okay, this is from my father. This is from my mother. Ni daripada ayah, ni daripada mak. Okay, this one and this one are homolog chromosome. This one and this ones are homolog chromosome. So kita tengok animasi tu berlaku macam mana. Exchange tu berlaku macam sana. Okay, this one we call as tetrad. Four chromatic. Okay, they are bind with 8 of certain protein. Saya tak nimal tadi. Okay, so along, oh my god, sorry, saya tertekan. Okay, so along the chromosome there are genes. Ada banyak genes lah, beribu-ribu gene lah. Okay, and then at the same level, okay, we call it allele. Okay, katakan this one dengan this one. Okay, we can find the name of gene are same. Example, this is gene to encode to make melanin. And if I go to the same level but at different chromosome, this level also we can find gene that encode to make melanin gene, but it is from different sources daripada individu yang berbeza, tapi nama gene tu sama. Mungkin gene tu exactly same, mungkin gene daripada mak dengan ayah tu ada perbezaan. Yang tu baru kita cerita pasal homozygote dengan heterozygote kan. Okay, so that is the concept of allele lah. Allele is gene that occupy at the same locus. Bila kita compare dua kita compare homolog chromosome. Okay, so crossing over happen, so they exchange, dia tukar-tukar lah. So that's why we can see there will be many version. Okay, mungkin dia jadi macam ni, mungkin jadi macam ni. Okay, mungkin jadi macam ni. It happen randomly. That's why Muhammad Nazif, if you compare with Khazani, it is not same. Dia tak sama lah. Sebab berlaku secara random. Recombination happen, so it produce a sperm or ovum with different composition. Dia ada variasi kat situ. Okay. Okay. So class, if crossing over do not happen during meiosis, katakan tak berlaku crossing over. If crossing over do not happen during meiosis. Okay, this one is from my father. This one from my mother. Warna dia lain dah sikit kat sini sebab saya jumpa video macam ni. Okay, from my mother has big B, big A. Big B, big A. And then this one ni dia replicate je lah. Dia replicate lah. That's why here is also big B. Here is also big A. Composition yang ni dengan ni must be same lah. Okay, this is different. Because this is from different individual. Copy that I get from different individual. Saya dapat daripada ayah saya. Okay, my father give me copy. Gene B juga, also gene B, but the version is small b. Okay, and here is small a. Dia lain lah, mak dengan saya ni lain kan. Okay, tapi gene tu nama dia sama lah. Okay, kalau this one gene B, this one pun gene B juga. Kalau this one gene A, this one is gene A juga. Okay, so okay, so if crossing over do not happen, okay, tetrad, okay, structure ni, okay, tidak berlaku exchange, so it will be separated. Okay, dia separate, dia akan separate juga. Okay, dia berlaku crossing over ke tak berlaku crossing over, dia akan separate. Okay, and then next division. Okay, so we get something like this. It remain, dia tak bercampur je. Kalau kat sini B besar, kat sini A besar. Kat sini B kecil, kat sini A kecil. Tak ada saya jumpa satu yang B besar, satu yang A kecil. Okay, why? Because crossing over do not happen. Okay, we try to watch another video here. Okay, if crossing over, okay, whenever crossing over happens during meiosis, cerita sama juga jin B dengan jin A. Okay, mak saya ada B besar, A besar, dengan ayah saya ada B kecil, A kecil. Okay, so this one happen. Ada berlaku meiosis. And then they are separated. Okay. First division in meiosis. Meiosis 1 dia ada division, dan it will be proceed with meiosis 2 ada division juga. Itu you belajar dulu lah. Okay, then we proceed with next division. Okay, so what we can see here, I can find big B, they are linked with small a. Okay, this one dia tak ada berlaku recombination. Okay lah, tak semua tempat tu berlaku, ada yang berlaku, ada yang bahagian yang berlaku. Okay, so this one, how is about this one? This one pun dia tak recombine lah kan, small b dengan small a. Tapi this one berlaku recombination, this one berlaku recombination, so it give variation. Okay, so without crossing over, okay, link allele segregate together. Dia akan segregate together lah kalau tak ada berlaku crossing over. The haploid cell contain the same combination of allele as the original chromosome. The arrangement of link allele has not been altered. Okay, ini sama macam dalam animation tadi. Okay, how is about when crossing over happen? Okay, kita dapat macam ni, yang kat atas ni this one these are term parental or non-recombinant cell. You have to memorize this word, parental or non-recombinant cell, means it is same like the parent cell, sama macam yang asal tadi sebelum di divide. Okay, but kita dapat we also get this one. This haploid cell contain a combination of allele not found in the original chromosome. This new combination of allele is a result of genetic recombination lah. So that one we call as non-parental. Non-parental maksudnya dia tak ada ikut macam mak ayah dia. Non-parental. Or we can say recombinant cell. So this is the source of variation. Inilah dia punya sumber kenapa saya dengan Khazani, saya dengan Hafizan, saya dengan Umi tadi, kami berbeza-beza walaupun kami menerima jin daripada ibu bapa yang sama. Okay. Okay, so clear cerita setakat ni. Ada sikit lagi. Bukan sikit lagi, ada lagi. Saya expect mungkin pukul 5 lah. Fit. Habis pukul 5. Okay, so far okay kelas? Before we move to another.
So far okay. Okay, doktor.
Okay, tak ada soalan, so saya proceed So there is story finding from these two important guy lah, dua scientist dalam bidang genetik ni. Okay, Peterson and Punnett. Bateson and Punnett. Cuma dia jumpalah satu benda yang dia tak tepat nak terjawab. Okay. Tapi yang bestnya dia jumpalah satu benda yang perlu dirungkai selepas itu. Okay. So siapakah yang merungkainya? Okay, Thomas Hunt Morgan. Thomas Hunt Morgan is an American evolutionary biologist or geneticist. Okay, he received Nobel Prize in 1933. Okay, because of his discovery about white-eyed mutation in the Drosophila melanogaster, lalat buah. Okay, dia jumpa something yang menarik bila dia kaji lalat buah ni. Okay, and of course it is related with linkages, genetic linkage. Okay, finding by Morgan, apa yang dia jumpa. Okay, the first direct evidence of linkage come from studies conducted by Thomas Hunt Morgan. Okay, so Morgan have provided evidence for the linkage of several X-linked gene. Okay, dia tengok dekat kromosom X, gene-gene kat situ nampak gayanya dia diwariskan dengan perkaitan yang rapat. Okay, close linkages. Okay, he investigated several traits that follow an X-linked pattern of inheritance in Drosophila melanogaster. Morgan's 3-factor crosses. Dia cuba kaji ada 3 gene merujuk melihat kepada 3 jenis phenotype. Okay, dan semua gene tu it is located on X-link, X chromosome. 3 genes that govern or control body color, okay, warna badan, eye color and wing length. Are all found on chromosome X. Semua dekat chromosome X. Okay, therefore the offspring tend to inherit the non-recombinant pattern of allele. Sebab dia duduk dekat chromosome X. Okay, the offspring, maknanya generasi yang selepas tu, bila kita buat kacukan tu, the offspring, they tend to inherit the non-recombinant pattern of allele. Okay, this one kena perhati betul-betul. Okay, we have 3 traits. We have eye color. Okay, it can be red, it can be white. Okay, we have body color, it can be grey, it can be yellow. Wing length, okay, it can be long, it can be short. Dia ada yang panjang, ada yang pendek. Okay, so this one, yang ni yang phenotype yang normal or wild type. What is wild type? Wild type means we can found it naturally. Dalam nature, kebanyakannya menunjukkan sifat tersebut. That is wild type. Okay, so kebanyakannya adalah Drosophila melanogaster with red eye, grey body and long wing. Mutant means species that undergoes certain unusual change. Dia ada berlaku change compared dengan yang asal. Okay, so dia ada mutasi. So dia menyebabkan warna mata dia jadi putih. Okay, badan dia jadi warna kuning. Dia punya wing dia jadi pendek. Okay. Okay, and then what Morgan mark this genotype as W plus. Plus mean here wild type lah. Wild type. Without plus means it referring to mutant. Tu dia punya cara dia buat lah. So we try to follow. We have to remember because for us to easy for us to understand the concept. So kita kena ingat jugalah, kalau plus tu maksudnya yang wild type lah. Kalau yang mutant yang tak ada plus. Okay, so this is the actual picture of Drosophila melanogaster with red eye. Okay, W plus. They are dominant. Yang tu dominant. W plus is a dominant. Okay, and this one W without plus, it's referring to mutant type. This is recessive. Okay, boleh setakat ni? Yang ni kena ingat, sebab yang ni kena ingat. W tu means it's referring to for the phenotype of the eye color. W tu merujuk kepada warna mata, sama ada dia merah ataupun dia putih. Kalau okay, W is referring to eye color, Y referring to body color, M referring to wing length. Okay, so kita bagi contoh 3 je, kita tak nak bagi yang 4, 5. Itu pening sikit kan, kita nak faham konsep saja, so saya ambil kat sini 3 lah. Okay, so if you find XX mean it's refer to female Drosophila lah, kan? Betul. If you find XY, this is male Drosophila lah. Okay, this one macam mana? Apa dia punya karakteristik dia?
Female tapi white type.
Okay, warna mata?
Merah.
Merah. Perempuan bermata merah lah. Okay, bukan perempuan, betina. Female Drosophila, red eyed. Okay, this one? Any volunteer?
Female Drosophila red eye.
Okay, why you say red eye? Because?
Dominant.
Dominant, good. Okay, this one?
Male red eye.
Male, red eye. Okay, this one?
Male white eye.
Okay, male, white eye. Okay. Kalau female white eye macam mana? Female white eye.
Chromosome
XW,
X
XW.
and W.
Okay, X, W, and then
X.
XW.
XW and XW. Betul?
Betul.
Betul lah, macam tu lah. XW dengan SW lah. Okay, yang tu adalah female white eye. Okay. Okay, this is experiment conducted by Morgan. Okay, this one female, right? Female yang bermata merah. Dia kacuk dengan jantan bermata mata apa?
Putih.
Mata
White.
putih. Good. Okay, so we have to separate them first. Kita separatekan dulu kan. Okay, and then we can combine it lah. Kita combine. As example, saya ambil pensel. Oh my god, mana pensel saya? Okay, this one combine dengan this one. This one combine dengan this one. So kita try to do the hybrid cross. Erase balik. Okay, so we get this one. Dapatlah macam ni. Dapat macam ni. Yang ini kan. Okay, this one is a female red eye. Betul. This one is female and this one is male red eye. Okay, this one female red eye.
Red eye.
This one
Male red eye.
Male red eye. Oh, semuanya merah. Semua merah. Okey, yang ni bapaknya mata putih. Maknya mata merah. Anak-anak tak kisah jantan ke betina semuanya merah. So from that clue, Morgan know, red eyes is dominant. Dia dapat maklumat macam itulah. Okay. So we try to breed F1 generation. Means kita ambil this one. We try to do we do the hybrid cross. Okay. Ataupun yang ni dengan yang ni pun boleh juga kan. Sama je lah. Sama je lah kalau ambil yang ni kahwin dengan yang ni. Ataupun ambil yang ni kahwin dengan yang ni. Sama je lah kan. Okay. So we get this one. Okay. This one dia separated kan. Ni dia separated. Lepas tu dia akan combine lah. Okay. Combine. So we get 2 red eye female. 1 red eye male. 1 white eye male. Okay. And then Morgan try to do the same thing. Buat lagi. Ulang lagi. Tengok. If the Drosophila is with white eye, it must be male. Semua male. Kalau yang warna putih je male. Tak ada jumpa yang color putih, mata putih, female. Okay. Kenapa? Okay, kalau mata putih betina macam mana? Dia punya genotype dia. W
XW, XW.
XW XW. Kan? Okay, how to produce XW from here? Nak gabung dengan yang genotype macam mana dengan yang macam mana? To produce this genotype. Kalau saya nak jugalah.
XW,
Kahwin dengan
Y.
SWY. Akan dapat ke yang red, white female?
Dapat.
Dapat.
Macam mana nak dapatkan XW dengan SW? Okay, apa tadi you kata? Apa kahwin dengan apa? Daripada genotype yang ada dekat sini lah. Genotype yang ada dekat depan mata kita ni. Macam mana kita nak hasilkan yang betina tapi mata dia putih? Nak ambil individu yang mana yang ada dekat depan mata kita ni?
XW plus and XW.
Okay, XW kahwin dengan SW kahwin dengan SW.
XW plus kahwin XW plus dengan XW. Okay.
Ni macam kahwin perempuan dengan perempuan kan?
XW plus XW kahwin dengan XWY.
SW plus this one XW.
sahaja.
Okay, XW saja. This one
XWY.
dengan Y.
XW dengan
Y.
Y. Okay. So kita akan dapat kombinasi yang ni dengan yang ni lah. Okay. So tu cara dia lah. Okay, kita ambil this one kan. Ambil this one. Kawinkan dengan this one. Betul? So dapatlah yang warna putih betina. Betul tak?
Betul.
Betul.
Okay. So tu cerita pasal mata sahaja. Okay. Sekarang kita, okay, from here, Morgan find that certain genes, it is X-link. Okay. Dia X-link. Dia provekan bahawa perkaitan genetic linkage is also need to be discussed in terms of X and Y chromosome. So this one, it is not elaborated by Mendel. So kita dapat clue lah bila dia gunakan Drosophila melanogaster sebab dia ada jantan dan betina. Okay, so Morgan 3 factor crosses involving 3 X-linked trait in Drosophila. Okay, 3 genes that govern body color, eye color, and wing length are all found on the X chromosome. So the offspring tend to inherit the non-recombinant pattern of allele. Sebab tu anak-anak dia tu dia inherit non-recombinant pattern of allele. Okay. So he tried to further the study. Okay, tadi dia tengok pada warna mata. Okay, daripada warna mata tu dia jumpalah bahawa concept of sex, X-link inheritance. Then he tried to expand. Dia try tengok jin-jin lain pula pada X chromosome juga. Okay, he tried to cross mutant female. Mutant female means it has white eye, yellow body, and long wing. Okay, dia cross dengan wild type male. Okay, dia try cross nak tengok apa jadi. Okay, and then he get F1. F1 he get like this. Okay, dia dapat wild type female, okay, with red eye, grey body and long wing. Okay, and then in term of mutant female, dia dapat white eye, okay, yellow body, and this is short wing. Dia dapat macam ni. Okay. And then what he do next is try to the hybrid the F1 generation, okay, to produce F2. And then he get this value. Dia dapat value ni. Okay. So kita tengok secara dengan lebih dekat dia punya apa ni, the data that he obtained. Okay. So he found that for this, saya ambil warna hitam. For this genotype, okay, nilai dia tinggi kat sini. And then this one tinggi kat sini yang mana ada yellow body, white eyes and small wing, dia tinggi. This one grey body, red eye, long wing pun nampak dia tinggi. Okay, so it tell us that this phenotype it follow male punya atuk dia. Okay, dia ikut yang male, this one. Dia masih lagi nak mengekalkan lah. Okay, and how is about this one? Ni dia macam nak ikut this one, female mutant. Okay, from the data it tell us lah macam dia ada nampak kecenderungan yang macam mana. Okay, Morgan observed a much higher proportion of combination of trait found in parental generation. Parental generation means non-recombinant pattern. So Morgan explained that all 3 genes are located on the X chromosome. Okay, so they tend to be transmitted together as a unit. Okay, And then we try to look closer. Okay, kita tengok dengan lebih apa ni, dengan lebih mendalam kan. We try to reorganize the data by considering the pair of gene separately. Kita tengok as example here, the first box here kita tengok perkaitan antara warna badan dengan warna mata sahaja. Warna badan dengan warna mata. And second table we try to look in terms of the color of eye with the length of wing. Okay, so first kita tengok dulu this one. Kita kaji perkaitan antara gene yang kodkan warna untuk badan dengan gene yang kodkan warna untuk mata dulu. Okay, it is here. You can see here. Okay, number here is very high. This one very low. So it tells us that this low number, it tells us that this is non-parental combination. It is rare lah. Okay, non-parental combination is rare. Yang ni pula common. Okay, so this one, ni nampak macam jauh besar lah beza dia kan. Okay, how is about this one? This one is fairly common to get this non-parental combination. Dia tak adalah macam ni, tak adalah macam data yang ke atas ni. Data yang ni this one dia reduce juga, tapi dia taklah rare sangat. Okay, it is fairly common lah. So the data tell us, what the data tell us, okay, the location of gene gene yang ikutkan untuk warna badan dengan gene yang ikut untuk warna mata, the location is close. From the data, it tell it is close. Physically, the location of these two genes are close. That's why we can get this kind of data. Nampak jurang kat situ lah. Okay, how is about the second table here? Yes, we also can see the difference here, but it is not as far as this one lah. Okay, so it tell us, this data tell us the distance of gene that encode for color of eye and the gene that encode for the wing. It is farther. Dia lah lebih jauh. Lokasi dia. Lokasi dia. Okay. When we say about location, it is subjective. Okay. Sangat jauh, sangat dekat. So that one is subjective. So that one can be interpreted by looking at the data lah. Data ni memberitahu kita tentang berapa jauh ataupun berapa dekat sesuatu gene, lokasi sesuatu gene. If two genes are apart from each other, crossing over is more likely to occur between that two genes. Compare between two genes that are close together. Chance of crossover to occur is very small. Okay, so data ni memberitahu something tentang lokasi sesuatu gene, sama ada dia jauh ke, dia dekat ke. Okay, so to explain further on this, Morgan consider the previous study by Jensen. Jensen dia buat study. Jensen, what Jensen do is he observe kias mata. Kias mata ni tempat di mana berlaku crossing over tu lah, point dia tu lah. Okay, and he proposed that crossing over involve a physical exchange between homolog chromosome. Okay, this is the actual picture. This is a diagrammatic picture lah. Okay, kita boleh nampak. Okay, so Morgan dia dengan cerdiknya dia perasan lah that crossing over between homolog X chromosome was consistent with his data. Dia perasan, and so he assumed crossing over did not occur between the X and Y chromosome. Okay, so the three gene not found, right, on the Y chromosome. So bila kita study sekarang ni pun betul apa yang finding ni. Okay, 3 gene tadi tu kita tak jumpa dekat kromosom Y. So daripada dia punya pemerhatian, daripada data, dia analisis, and he get very precious clue. Clue ni sebenarnya precious. Mengetahui kedudukan jin sama ada berdekatan ke tak berdekatan tu adalah precious. Kenapa? Sekejap lagi kita akan terangkan lah. Okay. Okay, so we move to next topic because how it is precious is connected with next topic lah. Kita ada dua topik kan. Dua kita punya aim yang kita nak faham. First, you have learned about relationship between linkage and crossing over. Okay, then second is you should be able to explain genetic mapping in animal. Genetic mapping is important. Okay, kenapa dia penting? Okay, so genetic mapping is also known as gene mapping or chromosome mapping. Its purpose is to determine the linear order of linked gene at the same chromosome. So when we do genetic mapping, we know the location of certain gene. Dia duduk di mana, dia duduk dekat dengan gene apa. Okay, so that information is very important for us to do the diagnosis. Okay, so you can see here, this is chromosome from Drosophila melanogaster. Okay, we have mutant phenotype macam mana, wild type phenotype macam mana, and then you can see certain number here. Okay, this number actually tell us about the location of certain gene. From this number we can know this gene and this gene, how close they are. This gene dengan this gene, how close they are, how far they are. So actually from the data, from this such data actually, it contribute to another data, which is this data yang di kat sini ni. Okay, data saya panggil ruler data lah, ruler, pembaris. Pembaris ni memberitahu kita something yang penting yang boleh digunakan, yang sangat penting dalam diagnosis, as example. Okay, okay. So genetic map actually are useful in many ways. Okay, they allow us to understand the overall complexity and genetic organism of a particular species. Kita dah tahu lokasi jin ini berada di mana, lokasi jin ini berada di mana. Okay, so they can help molecular geneticists to clone the gene. As example, we are going to clone certain gene. We are going to clone insulin gene. We need to know the location of insulin gene. Why we need to clone insulin gene? One of the important is we want to produce insulin protein by inserting that gene into certain microbe. So microbe tu dia akan melalui proses central dogma. Okay, ada transcription, ada translation, and it will produce protein. So protein tu it is important for people with diabetes. Yang mana yang kena cucuk insulin tu kan, kita cucuk insulin protein that we get from cloning technology. So to do the cloning technology, we need to know where is the location of insulin gene. Sebelum kita boleh ambil dia nak masukkan ke dalam certain microbe to produce insulin protein for us. Okay, they improve genetic map. They improve our understanding on the evolutionary relationship among different species. Oh, we know what is the connection between this microbe and this microbe, or this virus and this virus. Okay, this bird with another type of bird, penting dalam zoology lah. Okay, genetic map also can be used to diagnose and perhaps someday to treat inherited human diseases. Okay, nombor 4 itulah yang saya sebut daripada tadi kan, diagnosis. Kita nak diagnose penyakit, contohnya nak diagnose penyakit Alzheimer's disease. Certain genes, when it is mutated, it can cause Alzheimer's disease, example. So we need to know which gene and the location of the gene before we can do the PCR or whatever for determining the genotype of the gene. Okay, genotype of the sample. Okay, number 5, genetic map. They can help in predicting the likelihood that a couple will produce children with certain inherited diseases. It also can help for genetic counselor to predict whether they can get or the possibility of getting, the possibility of inheriting certain gene that cause the disease. By knowing genetic linkage, this thing can be in more proper way. Okay, so analisis akan boleh dilakukan dengan lebih terperinci tentang inheritance. Okay, genetic map provide helpful information for improving agriculture, important strain through selective breeding program. Okay, dalam kelas ni ada yang dietitian kan, program dietitian. So to produce certain food, we need to know this plant, when we breed with this plant, is it possible to get this kind of new plant? So to know this, we need to clear on genetic linkage. Okay. So genetic maps, it allow us to estimate the relative distances between linked gene based on the likelihood that a crossover will occur between them. Kita boleh estimate benda ni sebab kita tahu gene tu dekat ke jauh. Kalau dia dekat, mungkin dia akan inherit bersama-sama. Kalau dia jauh, mungkin dia tidak diwariskan bersama-sama. Okay, so experimentally the percentage of recombinant offspring is correlated with the distance between the two gene. Okay, kita boleh kira percentage recombinant offspring bila kita nak tahu distance between two genes. Okay, because we know if genes are far apart, kalau gene tu duduk jauh-jauh, we can get many recombinant offspring. Okay, if the genes are close, we will get very few recombinant offspring. Sebabnya masing-masing nak ikut bapak, nak ikut bukan bapak, nak ikut nenek, nak ikut datuk, dia punya phenotype dia. Okay, so this thing can be calculated. Map distance can be calculated using the data of recombinant offspring. So we try to get the number of recombinant offspring, divide by the total of offspring, so we times 100. So we will get the unit of distance. The unit dia kita namakan sebagai map unit, MU. Okay, and they also refer as centimorgan, cm, bukan cm centimeter, centimorgan. So you know how this unit, the name of this unit is proposed. Sebab dia sempena dengan nama Thomas Morgan Hunt tadilah. Okay, so untuk cerita pasal kedudukan jarak sesuatu gene, okay, kita guna unit centimorgan, bukan guna sentimeter. Kita guna centimorgan. How centimorgan or map unit can be obtained? By having this data, dapat data ni. Okay, And one map unit is equivalent to 1% recombination frequency. Okay. So, saya rasa saya mungkin exit sikit ni. Rasa sikit Saya kasih lebih. You ada kelas lain ke selepas ni? Tak ada kan?
Tak
Lebih
ada.
sikit. Tak ada kan? Okay, saya exit sikit lah. Pukul 5-10 minit pun okay, masalah kan? Dah nak pergi main-main lah. Main bola tampar ke. Main bola ke. Okay, ada sikit lagi So, so far okay ke? So far okay. Saya duk cerita panjang sangat, cepat sangat ni. Can you get the relationship between these two things? Okay, map distance dengan number of recombinant. Okay ke?
Okay, betul.
Okay, good.
Okay.
Kalau tak dapat catch up, you can, I will give this recording saya akan bagi recording ni balik. Okay. Genetic mapping experiment are typically accomplished by carrying out a test cross. Kita nak tahu ni lah tadi yang kita cerita pasal CM dengan MU tu, centimorgan dengan map unit tu, kita kena buat test cross. We have to carry out test cross. Okay, a mating between an individual is heterozygous for 2 or more genes, and one that is homozygous recessive for the same gene. This is the procedure. Procedure yang dah dijumpai oleh geneticist. Kalau nak buat test cross, kena cari macam ni, individu yang heterozygous for 2 or more gene. Okay, and another one is, and one that is homozygous recessive for the same gene. Okay. So figure 6.9. 6.9. Okay, this one Okay. Kita ada kita nak buat test cross, kita kena ada genotype yang sesuai lah. Okay. So this cross concern two linkage affecting bristle length and body color in fruit fly. Okay, kita cuba tengok contoh ni, phenotype bristle length. Bristle is bulu dia lah. Bulu dia tu panjang mana? Panjang ke pendek? Okay, bristle. Kita tengok bulu yang ada dekat kaki fruit fly tu kan. We will look at that characteristic, bristle length and body color, warna badan pada fruit flies. Okay, small s is for short bristle. S plus, ingat kalau plus it is for normal, normal bristle. Okay, kita ada satu lagi phenotype, E for ebony, body color. Ebony tu warna macam ni, warna putih kan, putih lah sikit. Okay, E plus is grey. Okay, E plus is grey. So yang plus tu maksudnya dia wild type. Okay, one parent display both recessive trait. Okay, it is homozygous recessive for the two gene, small s, small e, and then the other parent is heterozygous for the two gene. So kita ambil contoh S plus S, E plus E. Okay. So the S and E allele are linked on one chromosome. Okay, so S plus and E plus allele are linked on the homolog chromosome. Okay, so kita buatlah the hybrid cross. Okay, jumpa parent yang macam ni. Okay, kita buat the hybrid cross. Okay, dan kita bila kita dah cross, kita akan dapatlah offspring yang ada 4 karakter. Macam ni. Okay, this one the bristle is long and the body is grey. Okay, this one the bristle is short, ebony, the body is ebony, warna dia. Okay, so yang ni dia macam ikut balik dia punya parent dia. This one, dia ikut balik macam parent dia kan. So that one kita ada number of non-recombinant. Kita dapat sebanyak 537 untuk non-recombinant yang jenis macam ni. Okay, and then we get 542 non-recombinant juga. We classify as non-recombinant lah because it follow the parent. Okay, this one we get recombinant. Yang ni kita dapat recombinant. Okay, sebab dia long bristle ebony body. Dia macam campur kan. Campur karakter yang dekat atas ni. This one pun it is also recombinant. Dia campur karakter yang ada kat atas ni. So we have the number of non-recombinant. Okay, value ni. Okay, yang dalam kotak color biru, we have that number. Kita ada number tu, so we can put into the calculation. Okay, met distance. Okay, the formula is number of recombinant offspring divided by total number of offspring times 100. That is the calculation. The formula that introduced by the geneticist. Okay, so kita dah ada value. Okay, the number of recombinant offspring kita just take one. Okay, 76 and 75. Both of them are recombinant kan. Ambil lah total lah dua ni. Jumlah sekali 76 tambah 75. Lepas tu it must be divided by the total of offspring. Recombinant dengan recombinant kena campur semua. So we get times 100, so we get 12.3 map unit. Okay, so S and E gene are 12.3 map unit apart from each other along the chromosome. So dia dapatlah nilai jarak tersebut dalam unit map unit from the information of the number of recombinant offspring. Dia dapatlah jarak dia. Okay, okay ke kelas? Ada math sikit lah. Bukan math sikit, banyak juga math.
Okay.
Okay. Okay, so pukul 5 dah kita. So ada soalan? Semua okay ni? Bagus sudah UiTM di hatiku. Ada siapa-siapa nak tanya soalan yang tak faham? So slide ni saya akan bagi jugalah. I will pass this slide to Madam Hatini together with the recording. So if you interested to get the copy, you have to communicate with Madam Hatini lah. Okay. So kalau tak ada soalan, okay, thank you so much. So all the best for your next lecture session. Okay, this is ni awal semester ke, tengah semester ke, akhir semester ke, macam mana ni?
Tengah semester.
Oh, tengah semester lah. Okay. Okay, so okay, I stop the recording first. Stop recording supaya dia tak panjang sangat lah.